Hello, everybody, and welcome. So I would like to thank you for joining us for the second season of the Cyprus Biological Society e-lecture series, Excellence in Biology. And I am very happy to have here today Dr. Loukia Karacosta to start the season. So before we start, I would like to briefly give you an introduction to Loukia's research career thus far. So Loukia is currently an assistant professor in the Department of Cancer Systems Imaging at MD Anderson at Texas. She received her bachelor's degree in biology at the School of Biology at the National and Kapodistrian University of Athens at Greece. And then she did her PhD in pharmacology and toxicology at the School of Medicine and Biomedical Sciences at the University of Buffalo, New York. Before becoming a professor at MD Anderson, she was a postdoctoral researcher at 4Robin, which is a startup company that develops humanized antibodies with theranostic applications in cancer therapy. She was also a postdoctoral researcher at Cancer Systems Biology at Stanford University. She was a postdoctoral fellow there, to be more precise. And her current research interests include cancer systems biology approaches, single cell analysis, phenotypic plasticity, and therapy resistance in cancer. So, and I have to say that Loukia is one of the most hardworking and dedicated persons I know, and it's a pleasure and honor to have her today talk to us about her research. Loukia, the ground is yours.
Thank you so much for that kind introduction, Maria. And I thank the Society for inviting me. It's an honor to be here with everyone. And I will just go ahead and start sharing. Second. Can you see the screen?
Yes, yes.
Okay. All right, so I will be talking today about a project that started from my days at Stanford. And through this project, I had utilized cancer systems approaches, and I will be talking about what cancer systems is, to study in depth using single cell analysis, epithelial to mesenchymal transition, otherwise known as EMT, and drug resistance, specifically in lung cancer. Okay, so before I go into any details, I like to start with key terms and concepts that this study will be utilized in the whole talk. So to start with, we have cancer systems biology. There are many explanations to what cancer system biology is. I'll try to give you a very simple description. So with cancer system biology, what you try to do is to utilize multidisciplinary approaches to study a problem. So it could be cancer biology, it could be biochemistry, and then computational biology. But in the end, what you do is you use these multidisciplinary approaches to study the system of the problem. So not just the cell at the level of single cell, but then you work your way up to the organ and the system. What I'm really interested in studying is phenotypic plasticity and heterogeneity. So heterogeneity is a known issue, not only in cancer, but in many diseases. Nothing is uniform in the tissue. And phenotypic plasticity is this phenomenon where phenotypes are not driven only by mutations, which is what is the first thing we learn in biology. Cells have the ability to change their morphology and reactions and behavior, even if they're genetically the same with their neighboring cell. And this is a very dynamic process, and it plays a role in drug resistance and metastases in cancer. I will be talking a little bit about cell state transitions and trajectories. You will see some cool models and tools we use to be able to follow cells, even though we're not following them live, but you can infer the state changes of cells going from, let's say, epithelial to mesenchymal. And then in the end, all the data I will be showing today is single cell proteomic analysis through a technology called CyTOF. And again, I will be talking a little bit more in detail about what this technology is and what it has to offer. And I just wanted to say that if anyone has any questions, please don't hesitate to stop me and ask me during the talk. I have no issue with that. Okay, so what is EMT? So EMT is a process, and it's a developed, it's a well-known process that was first characterized during in the developmental biology. So, and through this process, an epithelial cell that is known to have tight junctions with its neighboring cells, and it has basal and apical polarity, goes through dramatic changes morphologically and biochemically, and it acquires a more mesenchymal phenotype that offers traits such as invasion, it's more migratory and more drug resistant. Now, this process is very dynamic and very plastic, and it is known that it can also be reversed, and that's called mesenchymal to epithelial transition. Now, in cancer, MET matters because it is hypothesized that when cells enter the bloodstream to metastasize, they're in a mesenchymal phenotype. But then when they reach the distant site, they undergo the reverse so they can start proliferating again and form a new tumor at a distant site. Now, in cancer, it has been linked to metastases, drug resistance, stem-like features, and poor clinical outcome, especially in the lung cancer field. So in the lung cancer field, which is what I will be talking about today, it has been linked to tyrosine kinase inhibitor resistance, tumor immune evasion, and then recurrence. So there's been signatures of EMT transcriptomic levels that link the patient's probability to recur or not. I just want to bring your attention to these, the cells in green. These are called partial EMT. So overall, EMT is not a binary process. You don't start from only epithelial and then all of a sudden become mesenchymal. You actually go through different steps. And the partial EMT means you are co-expressing both epithelial and mesenchymal features. And the reason I'm mentioning this is because these cells are what are most observed in clinical specimens, and they are considered to be the most plastic and clinically significant. Okay, so let's now go into what is mass cytometry, other known as CyTOF, cytometry by time of flight. So really, when I explain what CyTOF is, it's really a fancy type of flow cytometry. So if you know what flow cytometry is, you get measurements of single cells. But because you use fluorophore antibodies in flow cytometry, there's a limit to how many markers you can interrogate because there's spectral overlap. So now there are even better technologies. Maybe you can go up to 12, but there's always a limitation. Now with mass cytometry, what you have, sorry about that, is your antibodies now are not linked to fluorophores, but they're linked to metal isotopes. So there's less overlap, and thus you can use more antibodies at the same time. So theoretically, you can actually probe your sample with up to 100 different antibodies. You don't need to compensate like flow cytometry. There's not much background. And then you can get from millions of cells readings of 100 proteins at the same time. Now most people do 30 to 50 proteins, but like I said, it has a capability to measure 100 proteins per cell. So you can imagine if you can build an antibody panel of around 50 antibodies, you can then start interrogating at the same time per cell different types of cellular processes. You can do cell proliferation, apoptosis, metabolism, anything that is what is interesting to your research and beyond. Now, where it gets complicated is actually the data analysis. I won't get into too deep computational methods, but overall, once you get the raw data and you transform them, what happens is you have a high dimensional data set. So it's hard to do biaxial plots for 30 proteins for 1 million cells at the same time. So what ends up happening is you do the dimensionality reduction so you can actually visualize the data in 2D dimensional space. And then you start by using the most important markers, and I'm sorry this is moving. You do clustering, and then you try to visualize and interpret the data. Okay, so the goal of the experimental approach was to study EMT and try to see the link with lung cancer, for example, drug resistance. But the problem is that EMT, like I said, it's not binary. It's a spectrum of many intermediate states. And up to recently, it hadn't been well defined at the single cell proteomic level. If you're able to do that, then you can start understanding more its link with resistance at the single cell level and see the heterogeneity. And thus, you can start building reference maps so others can use because you've already characterized it. So overall, what we did was we took lung cancer cell lines, we optimized the treatment to induce EMT and MET, specifically by using TGF-beta, which is known to induce EMT, and took different time points, then induced MET by withdrawing TGF-beta, and then we took all these time points together and we stained them with around 30 antibody panels shown here, interrogating different processes, not only markers that characterize E and M states, but also signaling and stemness and others. And then once we got the data, the goal was to build a reference map of what we had defined of different EMT states. And then you can imagine that if you have a reference map from lung cancer cell lines, you can then project when you analyze clinical specimens and assess, okay, what states of EMT exist in my specimen? And is this linked to either poor clinical outcome or drug-resistant features? And also in parallel, we also interrogated how the cells move from one state to the other, shown here. And you're going to say, okay, that's a fancy analysis, but why does it matter? Well, if you know where it's most probable for the cell to go, you might be able to predict resistance and maybe fight it before it arises. Okay, so the first thing we did, because I wanted to characterize the entire spectrum of EMT from start to beginning, we had to choose a cell line that actually starts very epithelial and becomes very mesenchymal. So these are three very well-established lung cancer cell lines in the field: A549s, H3255s, and HCC827s. And we first just did a morphological analysis and using Western blotting to just do bulk protein analysis of markers that are very commonly used for EMT. So just briefly, E-cadherin is a very well-known epithelial marker. It's a marker that will be expressed at the surface of cells, and it keeps them signaling to each other and connected. And then vimentin is a mesenchymal marker. So when you're undergoing EMT, you should be starting to lose E-cadherin and gaining vimentin. CD44 is also a known mesenchymal marker that is supposed to go up with EMT. Now, A549 cells, you can see at basal levels, they have a very epithelial phenotype. And here I'm showing you a 10-day treatment with TGF-beta. You can see cells dramatically change morphologically. They become elongated, spindle-shaped, fibroblast almost. And you can see from the protein analysis that they are already at a partial EMT state. They are co-expressing both E-cadherin and vimentin. And then with treatment, they lose E-cadherin and just become more mesenchymal. This cell line, to the contrary, they start very epithelial, but they barely change with TGF-beta. So this is around 2 weeks. And all we did was just a little bit of vimentin at bulk level, and that was it. So these two cell lines didn't really give me the opportunity to say, okay, I'm going to study the whole spectrum in lung cancer cells. 827 cells, they start very epithelial. So you have E-cadherin, no vimentin or CD44. They dramatically change, and then they acquire high levels of vimentin and CD44. So here I'm just showing you a nice immunofluorescent image of these same cells. You have control, red is E-cadherin, very epithelial, and then they acquire high levels of vimentin. And just notice the heterogeneity of the shape of cells. You have large, very small, elongated, and then there's a lot of heterogeneity, and this is just the morphology, not the protein expression. And just an interesting aspect that if you have cells without TGF-beta and they're very sparsely seated, they automatically turn on EMT. So this is with no TGF-beta. So if cells are left kind of alone, it's like it's an autopilot and they just have to switch to become EMT, and maybe it has to do with survival mechanisms. So the first thing we saw that it was spatially dependent. Now, before you do CyTOF, because it's very similar to flow, first you do some optimization with flow cytometry, see if everything works, see if you see what you would expect. So here I'm showing you biaxial plots of E-cadherin and vimentin and CD44, CD24. And you can see the cells with EMT, they acquire vimentin, high vimentin there, and they lose it with TGF-beta. And then they also, they lose the E-cadherin while acquiring vimentin. And then CD44-24, and these are markers that are used for stem-like characterization. You have an increase of CD44, and you're supposed to see a decrease of CD24. So in the field, when you have high CD44, low CD24, you're considered to be stem-like, not necessarily a stem cell, but stem-like. But related to the phenomena that you have to have them spatially sparsely seated, you can see that if I do a 2-week treatment and during that treatment, I continuously reseed the cells without letting them become too confluent, you see an even more EMT signature and protein expression. So you can see that the cells dramatically, if I had reseeded a few days before during TGF-beta treatment, you have high vimentin and low E-cadherin. And then CD44 goes even higher, and you start to see that movement towards the left, the CD44 high, CD24 low. So just from the flow cytometry and some manual gating, you can see that there's pretty much 4 states. You have the very epithelial number 1, and then cells become more partial, more mesenchymal, and then a small population becomes mesenchymal stem-like. But since we have CyTOF, you can understand that there's a lot of heterogeneity between these states. So then by building the 30 antibody panel, you can start interrogating the heterogeneity within these states and start mapping out the process. So the plots I'm showing now, which are again E-cadherin and vimentin, these are now CyTOF data. So they look very much like flow. So our technique is working, and you can manually gate the 4 canonical states we called of EMT. And you can see the cells with TGF-beta at 2-day, 6-day, and 10-day, they're becoming very mesenchymal. Now, what happens when we withdraw TGF-beta? Now you see the cells, they become more epithelial, like you would expect, not necessarily all of them within the 10-day time frame. And then the plots show you that the proteins will start to go back to where they started. But this is just the 4 markers were discussed. Like I said, we had around 30 markers. And then this is just showing you every marker with time, how they can dramatically change. And then you can see the heterogeneity. Now, this is just distribution of the cells with time. It's not even showing you the subpopulations, but you can beautifully see the dramatic drops in E-cadherin. Vimentin goes highly up at the 10-day, and then it starts going back at the 10-day withdrawal. W is withdrawal. Dramatic increases in CD44. Twist is a transcription factor known to drive EMT. It's interesting that you don't see all the cells expressing it, just a subpopulation. And it's very dynamic. It goes up and then down. And these things you would miss if you didn't do single cell and a nice timed course analysis. And then there's other epithelial markers that play a role, signaling markers, cell cycle, and PD-L1 that is very important in cancer because this is actually a targeted molecule. And if the cells are expressing high of PD-L1, they can evade the immune system. So we just confirmed here that it does in fact go up during EMT in lung cancer cells. Okay, so now we go a little bit into the computational analysis, and this is with help with other computational postdocs in the lab. We worked together to see what was the best method and the best visual representation of the data. So here, this is just a simple clustering analysis. So what you do in very simple terms is you take all the cells from all the time points and you put them together, right? So what the algorithm is going to do, it's going to find the most dramatically changed markers and then cluster all the cells in those clusters based on the expression of those markers. So our analysis showed that the most efficient clustering markers were E-cadherin, vimentin, as expected, CD44, CD24, and they're all shown down here, twist and MUC1, which is an epithelial marker. And you can see now the heterogeneity of the cells per state. So we were able to identify 3 epithelial states, 3 partial EMT states, a mesenchymal state, and an MET state, so on the way back. And what the plots are showing you is this is the level of expression of each protein, and every little bar is a single cell. So you can see that, yes, you can have a uniform, they belong in these groups of cells, but even within there's some heterogeneity. So for example, in the partial EMT states, there were subclusters of cells that were expressing the twist transcription factor, and you would have missed that if you just did bulk analysis. So briefly, just to show you, epithelial states, as you expect, high E-cadherin, low vimentin, and then there's variability in other markers such as MUC1. If you have partial EMT states, as expected, you have high E-cadherin, high vimentin at the same time. Mesenchymal state, you completely lose E-cadherin, you have high levels of vimentin, and then the MET state, which is a very interesting state that came up in our analysis, and we were actually the first to identify an MET state at the single cell proteomic level. It's interesting because it still is very mesenchymal when you look at E-cadherin and vimentin, but it also has high MUC1, which is an epithelial marker. So it's a switch of them returning back to the epithelial state. Now you're going to say, okay, how did you define this as MET? Well, don't forget, we also had time information. You would expect that epithelial states are highest at time 0, partial EMT states in between. Most mesenchymal cells should be at the 10-day, which was the maximum time in TGF-beta, and MET should appear when you start withdrawing TGF-beta. So as you can see here, these are number of cells per state during the time course. So you can see epithelial states highest at time 0, and they go down. Some of them return after withdrawal. Partial EMT states are in the middle of the time points. Mesenchymal state, as expected, the highest at the 10-day TGF-beta. And then the MET state, it only appears once you start withdrawing. So the spike here is at the 2-day withdrawal time point. Okay.
Loukia, we have a question from Dr. Pablo Neofitu.
Yes.
Pablo, I'm going to give you, you can directly give the question to Loukia.
Yes, please.
Yes. Is it right that only about half of the cells go back from mesenchymal to epithelial after removal of TGF-beta?
Yes, very good observation. So within the time course we did, maybe it was a little bit more than half, but yes, not a big proportion of them did not return. Now there can be different explanations for that. Maybe if we left them longer than 10 days, they would have. And the only reason we stopped at 10 days was because we wanted to match it with the days in TGF-beta. However, literature says that some of the cells, once they become mesenchymal, they pass this epigenetic state and they get fixed and they might not be ever able to return. But I just want to say in the context of cancer, you do want the cells to be able to switch, because if they don't switch, they won't be able to colonize a new site. So that, yes, that is a good observation. We didn't do longer times of withdrawal. And we can't know for sure if there was an epigenetic switch that fixed some cells in the mesenchymal state.
Thank you.
You're welcome. Thank you for the question. Okay, so now I'm going to show you a different way of visualizing the data, and it's a very cool way. So there's many tools out there to visualize and cluster high dimensional single cell data, and this is just one of the ways. So here I'm showing you a force-directed layout of the cell events I was just showing you with plots and biaxial plots in the graphs. So here, every dot is a cell, and the closer the cells are to each other, the more similar they are in their protein expression. And then you can color with this program depending on the parameter. So it could be time, or it can be a specific protein of interest. So, and we were calling this the EMT bird, because I don't know if you see it, but it does look like a bird. So you can see that, for example, here I'm coloring with time. So blue is your time 0. These are the intermediate states. At the middle is 10-day TGF-beta. And then the more red states are the cells that are undergoing withdrawal, and they return back to here. Now, this is also epithelial. The only reason it's being separated from here is because we're using the time parameter. So it's a difference, and it pushes the cells apart. So you can see a nice representation of all the cells and how they may be connected to each other in high dimensional space. Now here I'm just color coding each of the states that I discussed with you. So you have the 3 epithelial states, partial EMT states, mesenchymal, MET state on the way back, and then some of the states and not all of the cells we're revisiting, E1 and E2, for example. Now this circle here with the asterisk, these cells here are those stem-like cells that I talked about, the high CD44, low CD24. And because they were so small in numbers, our clustering algorithm did not pick it up as a different cluster. But because we can see each protein at each spot, we can actually manually characterize them and define them as a state. So this is what I'm doing here. Here you see now in all this space, every protein that might be of interest to any researcher. So you have high E-cadherin, it drops and it starts going up on the way back. Vimentin should be the reverse. CD44 is like a mesenchymal marker, so it looks like vimentin. There's heterogeneity of CD24, but right here is that circle of CD44 high, CD24 low. And then MUC1 is the interesting marker that I talked about, that it's heterogeneous in the epithelial stage. Although it's an epithelial marker, it goes down, and then it's the first epithelial marker to come up when the cells start switching. And then TWIST, which is an EMT specific marker, you would expect them to be highest at the most mesenchymal states. But no, with our analysis, it actually comes up in the partial states and then it goes down. And actually, I did find a paper that showed that TWIST, although it drives EMT processes, it only goes up during, and it's supposed to drop before cells acquire mesenchymal stem-like states, which agrees with our data. Now, if you go into the paper, you can actually see all the other 30 proteins and how they are within this high dimensional space. Okay, but like I said, the primary goal was to actually build a reference map of all these states. So it's fancy to look at the nice bird-like shapes, but I wanted something to have some translational applicability, because if a doctor gets analysis of tissue, and I know we're far from that step, but let's say he does, and he wants to see, okay, is this tumor epithelial? Is it mesenchymal? It's hard to see it every time you have a nice fancy shape with single cells. So I wanted to build a map to then, when we have it, every time you analyze a new sample, you can project it on it, and then you can see where the cells fall. So with the different type of analysis, we took the data and put them on a different 2D plane, which is t-SNE here. And again, it's the same data, but then we identified the highly dense areas of the cells and broke it down into maps. So basically, every area of the map gives you the highest probability or confidence that your cells belong to that state. And then you can again look at every marker within this reference map. Epithelial states, ECAT-herin, partial, it drops and it's still low at MET. The menton is the reverse. TWIST, you only see it in the 2 partial EMT states. CD44 with the circle of the stem-like cells, and then the MUC1, which is a very interesting marker. Okay, now does it work? So this is showing you again the bulk analyses of the other 2 cells that we talked about, the cells that were already partial, they become mesenchymal, and the cells that barely change from their epithelial status. I took these cells and these time points, I did the same CyTOF analysis with my 30 antibody panel, and then you project on the map. And you can see that in fact, A549 cells are on the partial states, they become more mesenchymal with TGF-beta. And then the H3255 cells, they're mostly epithelial and they barely change. So it's working in terms of, can you have a general assessment of the EMT status of a cell line? And here I'm just showing you the biaxial plots if you had only done ECAT-herin and vimentin and CD424, confirming the changes you would see if you just looked at the map. So they're very partial, they become mesenchymal, and then these cells, they barely change from their epithelial status. So what we wanted to do for proof of concept is now take some clinical samples. So these were primary resections from lung cancer patients, dissociate them, stain them with our same panel. But now in our panel, we included antibodies to separate immune cells with CD45, endothelial cells with CD31, and FAP that separates stromal fibroblasts. And here I'm just showing you that with these antibodies, with their expression, you can actually separate what we assume to be tumor with the cytokeratin levels in malignant cells. So what we then did is we gated out the tumor cells from the tumor microenvironment cells, and we projected on our map. And now you start to see the spectrum and the heterogeneity of states that might be visited or exist within an individual tumor of a patient. So, and then you can also start correlating with their mutation. So for example, in the literature, EGFR mutated tumors, which is, it's very commonly mutated, they tend to be more epithelial. And we did see that right here. And cells that acquired additional mutations or were KRAS, they started to become more mesenchymal. And here I'm just showing you a chart of the percentages of cells within each state. Obviously, this is not a big number of samples. You can't make any statistical correlations. It's just a proof of concept, how it might be utilized. And it's interesting because, for example, the A549 cells, which are also KRAS mutated, this is where they fell in their basal state, if you remember, right here, right? But the 827 cells, their basal state is more epithelial, and you can see it here in the tumor, these tumors as well. And then the other thing we noticed that from every tumor, we had our CD45 cells. We noticed that the more infiltrated it was, the more mesenchymal it was. And this does agree with the literature because CD45 immune cells are supposed to be a source of TGF-beta. So that might be the explanation. Note that I didn't really see any significant numbers in that MET state. And I'm not showing it here. We analyzed 5 more clinical specimens. We never get a significant number of cells on the MET state. Now, you can argue that makes sense because it's supposed to happen later once they metastasize. So maybe this agrees with that, but I'll have some interesting data to show you about that as well in subsequent slides. Any questions? Because I'm going to be slightly switching to other applications. Okay. So what I was really interested in was drug resistance. I think if we can figure out a way to predict what the cells are going to become with drug treatments and then treat that, we might do better in the clinical world. So it has been shown that many drugs in the clinic actually induce non-genetic heterogeneity. Now, this is different than selecting a pre-existing mutated resistant clone. This is more dynamic and short-term. You can actually see that when you treat your cells in culture, for example, they can immediately acquire EMT-like features, and that will give additional resistance. So here, I'm treating the same cell line with erlotinib, which is a tyrosine kinase inhibitor. So it inhibits mutated EGFR, which is a very commonly mutated marker in lung adenocarcinoma. And you can see that cells are changing morphologically. I know this is a little bit small to look at, but you can see that but it's different than the TGF-beta. In the TGF-beta, there was like a spectrum of many different morphologies. In this case, it was two. It was either very mesenchymal, the resisting cells, after 2 and 21 days in the drug, and then cells that looked very epithelial. And it was a very interesting observation. I only was able to get a 5-day withdrawal, and you can see that there's less mesenchymal cells there. So I'm just circling the morphologies that we saw in the resistant cells. Because it was shown that erlotinib can induce stem-like features, that was the first thing I looked at. So I analyzed these with CyTOF, and here I'm just showing you the CD44-24 plots. And you can see that from 0.26% at the 21-day, it goes up to 4.33. And then even with just 5-day withdrawal from the drug, you can see that number drops. So this did confirm some studies that were already out there. But what I really wanted to do was to see if the map works in terms of drug-resistant phenotypes. So here you have your control cells, very epithelial, just like our TGF-beta experiments. And then when we added erlotinib, you can see that there's a significant density right where you expect the mesenchymal-like. But at the same time, cells are becoming both mesenchymal and epithelial. So it's not only stemness that confers resistance. There could be some epithelial states, this is where heterogeneity comes, that because of altered signaling can confer resistance even though you're blocking EGFR. And then I'm showing here that with withdrawal, the cells move towards where they were before. Showing you the differences here. And then just to give you an example of that, even epithelial different states can give you resistance. I'm just showing you E-cadherent. So we're inducing more mesenchymal phenotypes and stem-like, but in the epithelial states, there's an overall increase, more homogeneous high E-cadherent than when before you added the drug. And that might have to do with increased other signaling pathways that confer resistance to all these phenotypes that you see here. Okay, now just a small shift. Like I said, we also wanted to interrogate dynamics of cell state transitions. And here I'm showing you, it's the same data. It's the density plots on the map of every time point. And now you can see that the cells start here and they go from left, the partial, they reach the mesenchymal. And at this point, it looks like, and this is where I was saying that there may be a switch. Some of the cells will undergo the MET and cells that are here will just go back the same way. So I don't know if you remember when I said that partial cells seem to be the most plastic. This might be linking to that phenomenon. So it looks like if you were in a partial EMT state and you take away the stimulus, you're able to go back the same way. But if you pass the mesenchymal threshold, you go through a different route. Now, this is very interesting in the field of EMT because there were studies that were showing with theoretical models and just gene transcription data that there's this phenomenon called hysteresis, which is a phenomenon that the future state depends on the previous one. And you might go back to where you came from through a different route. Now, this is a very like cool theory and phenomena that we just wanted to see. And it happened that we actually confirmed it with single cell proteomic data. And like I said, it matters because if this is validated, and in fact, we know the transition probabilities and you know where the cell is going to go with treatment, you might be able to treat it beforehand. Here, again, this is just some nice computational analysis that we actually got transition probabilities from state to state. And I just want you to focus on the thicker lines. You know, you see the movements from here, and then you actually see bidirectionality. So you see EMT2 can actually go back to PMT1, for example. So it's showing you that plasticity. And then this was the most interesting phenomenon because it actually shows you mathematically that yes, there's the M and they can go to MET. So again, this was just an interesting side project that we did with computational scientists at Stanford. Okay, and then like I said, and I'm not going to go any further to this, this is why this is interesting, because if you can interrogate trajectories among drug persistent states, then hypothetically you can beforehand treat for that or predict it or just keep it from happening, hypothetically. Okay, so the last part of the talk would be a project that was funded a little bit after we completed this EMT story. And the idea was, okay, you know, the problem with clinical specimens, you can't get all the time. You can't get time resolution. It's hard to get multiple biopsies, obviously. So it's hard to see the dynamics of EMT and MET in clinical specimens. You can just get a snapshot. But we wanted to see if, for example, we can ever detect the MET state, which is important in metastases. So if we were able to get multiple samples per patient, can we really assess disease status and progression at a personalized level? And do we see any correlations with immune populations, especially now that the whole world is into the immune system and how we can utilize that to fight cancer, right? So this project now was a little bit different. The idea was to obtain pleural effusions from lung cancer patients. Now, pleural effusions, what they are is, especially in late stage lung cancer patients, but other cancers too, because of inflammation and tumor burden, there's an accumulation of fluid in the pleura of the lungs. And many times there's tumor cells within the pleura. Now, literature shows that is also a way of metastasizing. It's called transcoliomic metastases. And in general, if you have pleural effusion built up, it's a very poor signal for your progression. It's usually in late stage. It keeps them from breathing well. So pleural effusions are actually taken all the time out from the patient because it keeps them from breathing well. So it's a good biological specimen to see, do I see any dynamic changes while they're treating the patient? Because there's actually tumor cells within the fluid instead of doing surgery and resecting the patient, which is not, you know, that's not feasible. So what we did was we obtained pleural effusions from late stage lung adenocarcinoma patients. And then we run our CyTOF analysis, but now I included even more antibodies to my panel to now detect different immune populations. And then after we ran CyTOF, do two things: see the landscape of immune cells in the lung cancer patients, and then at the same time, take those tumor epithelial cells and project them on the map and see where they are in EMT status. So here I'm just showing you that we had 8 patients. 2 of them, their pleural effusion did not have malignant cells in there. And then the rest of them, we did detect even with the pathologist cytology report. And then all of them were undergoing some form of treatment, except one, which is colored in blue. And then, like I said, now I'm going to be showing you this type of analysis on these patients. The cool thing was that one of these patients, I was able to get 2 samples while they were going treatment for 2 months. I was hoping to get more samples per patient, but because of COVID and everything shut down, the project stopped there. Okay, so here, this is again a force-directed layout like I showed you before. We took all the samples and we did the clustering, and this is just looking at them at high dimensional space. And I'm just color coding the different cell types you see in all the samples at the same time. You have your CD8 cells, CD4 T cells, you have NK cells, B cells, myeloid cells, and then in red is when we were detecting cytokeratin positive that we were assuming were malignant cells. And the first thing we saw was a correlation between tumor cells and myeloid cells. So here I'm showing you, this is having all the patients together except the ones that we found tumor cells in. And you can see that if there was no tumor cells in red, you also didn't have myeloid cells. But if you ran just the samples that had tumor cells, red, you also had myeloid cells. So basically what I'm saying is there was a correlation between myeloid cells and cytokeratin positive cells in pleural effusions. If you detected a large number of CD33 positive cells, it was almost sure you were going to also detect cytokeratin positive cells. And here I'm just showing you again, every patient's distribution of immune cells in total and individually. And you can see again here that the only patients that had red, which is the percentage of tumor cells, were the patients that had myeloid cells. But what we were really interested in was, okay, if I take these cytokeratin positive cells, where do they fall on the map? So again, these are a small number of patients, but it was still interesting to observe. Here we saw two things happening. You either had tumor cells falling on the partial EMT states or tumor cells falling on the MET states. So this is all we saw. And this is the first time we observed MET state. Remember, I told you we saw it in the in vitro data. We didn't see it in the solid tumors, but in the pleural effusion, there were high numbers of MET state. And if I wanted to see if there was any correlation or relationship with treatment, so this patient was our treatment naive, and it was primarily at the partial EMT. This patient had a history of treatment, but he hadn't been treated for around 2 or 3 months, again, partial EMT. The patients that were undergoing treatment were primarily projecting on the MET state. But let's focus on here now. This patient here was a patient that I was able to get 2 samples. And we saw an interesting phenomenon. You can see that with the treatment, cells are moving towards either the stem-like area, if you remember, or the partial. And I'm just showing you the increase in the stem-like cells. And why it was interesting was because this was very similar to what we saw to the cell line data. And osimertinib is again an EGFR inhibitor, just like erlotinib. So it was just cool to see that it's not a confirmation, but at least something similar to what we were seeing in our in vitro data. So overall with this project, which we're trying to publish soon, is that it's the first time that you actually see the MET state in clinical specimens, and it might be related to metastases and therapy. I just want to note that the only patient we got absolutely no MET cells was a patient that had no metastatic disease at the time. So like I said, MET matters for metastases, and at least from 5 patients, I know this is not significant, we saw that, and it was very interesting. And it's just, again, a proof of concept. Such a map, if we were able to build even better maps and informed by more clinical samples, it could be used in the future to assess therapy response and resistance at a personalized level. And then what's missing from all of this and future plans of my lab is actually now seeing, not dissociating the tumors and getting the spatial-temporal relationships of what I showed you once you dissociate the cells and stain them. So now we're lucky because there's many cool technologies that you get the same multiplex staining on tissue now. So you don't dissociate your tumor, you don't lose cells, and you actually get to see all these phenotypes in space and the relationships of cell types and their heterogeneity amongst them. And I'm just showing you, this is from another paper. There's different technologies. There's technologies similar to CyTOF that you use metal antibodies, or there's fluorescent technologies where you wash and restain, and you can stain up to 50 proteins. And here I'm just showing you that once you stain your tissue and you see all these cellular types, then you can start making neighborhoods of cells. And you can say, oh, I always see a neighborhood of a CD4 T cell with an M cell, a mesenchymal cell, and an endothelial cell. And this is correlated across patients with poor survival, for example. So this is some very cool analysis that you can do and link with the CyTOF analysis. And, you know, at the end, it will be cool to just map the tumor microenvironment landscape, not just the cytokeratin positive cells. So in ending, I just want to say that this was my work before coming here, and I'm trying to build on it. But the whole idea is to use cancer systems biology approaches where you use both in vitro cells and you build up with the data you're able to get and control. And then you can also, from the top bottom, you can get tissue and analyze that and go down to the single cell level. And with system approaches and computational analysis, you can bring it all together and study the system and be more clinical or translational. So for example, a thing that my lab is focusing now is to perform different treatments that are used in the clinical in cell line spheroids or organoids, so you at least be a little bit closer to what's happening in vivo. And in that case, you're not just treating with a drug in an isolated cancer cell line, which is what most do. You're also at the same time getting phenotypes and heterogeneity of the fibroblasts, the endothelial cells, or the immune cells, and you can map them out. And then you can confirm if those mappings relate to their neighborhood spatiotemporal relationships. And with all this data together, you can imagine that with collaborating with statisticians and computational biologists, maybe we can start getting closer to predictive features of therapy response and survival in not only lung cancer patients, because you can understand that all these things I talked about are applicable to any cancer. And that's where we're hoping to go in my lab. And with that, I want to like to thank you for your attention and thank a lot of my past mentors and collaborators, the grants I was able to, lucky to get while a postdoc, and the funding here at MD Anderson. And I'm happy to answer any questions. Thank you.
Thank you, Loukia, for this wonderful and very detailed presentation. I must say my favorite was the bird-like shape.
Yes.
and the codex. The codex
yes.
Is amazing.
Yes.
So I would like to say to everybody, if you have any questions, please raise your hand and I will unmute you so you can directly ask Loukia your question. So until somebody decides for a question, I have some. So I was wondering, oh, wait, somebody, so let's see. We have in our company, Professor Konstantinos Zeltas. It's hello, Dr. Zeltas, and you are free to ask your question.
Can you hear me?
Yes.
Well, actually, I don't have a question. I was kind of overwhelmed with this excellent presentation.
Oh, thank you.
And I had to take care of other things as well, so I couldn't attend to the level I would have liked to. But in any case, I would appreciate if you contacted me at the email I gave.
Okay.
So that we exchange some information and get to know your group better, and you get to know our group.
I would be happy to. Yes,
yeah,
for sure.
I believe you're a young, very promising Cypriot scientist abroad, and we would like to see if we can explore your capabilities.
Oh, definitely. I will be happy to. Thank you.
She is indeed one of the best.
Well,
so, and Professor Zeltas has created the Cypriot Biobank. So, yes, so you are in good hands, very good hands. I will bring you in touch with the end of the meeting.
Very important to have good biobanks. It's
yes.
Yeah.
So we have a question from Dr. Neophytou. The floor is yours.
Yes, Loukia, congratulations on the excellent talk.
Thank you.
How near are we to a clinical application? I have noted that you already did some studies with the pleural fluid for lung cancer patients. Will there be soon a clinical application, for example, for lung cancer?
That is an excellent question. Okay, to be honest, We are not as near. This is all very proof of concept. So we're trying to submit this paper to see the feedback. Now, being at MD Anderson, I'm very new here. I'm hoping that with the collaborations, maybe we'll be able to submit a grant and actually get big numbers of pleural effusions and see if this is really applicable. I do have faith in it because it gives you both a single cell heterogeneity. It gives you a way to assess in general, but it also gives you the personalized aspect, which is the future of medicine. We all talk about it, but nobody does it. So I am hoping that being here at MD Anderson, but to do that, we would have to find a pulmonologist that believes in the project and put a grant together and get many samples. So the limiting factor is to get number of samples. And because it's pleural effusions, yes, we might be closer than with solid tumors because I would be hypothetically able to get many. Like I said, I had got all those samples and then COVID started and we shut down. And then within that shutdown, we moved from Stanford and I was never able to make it a little bit more, broad and in numbers. But I think the promise is there. The other issue is, yes, CyTOF is perfect. It's beautiful. It is expensive though. So I would assume that, MD Anderson has a big core and they can do it, but in order for it to become clinical practice, there must be money and, funding for it because it's not that cheap to run a CyTOF panel. But having said that, if you notice, even though we got the beautiful biology and characterization, we did nail it down to 6 markers in the end to build our models. So since we did all the single cell analysis and we get nice, beautiful biology, in-depth insights, but we also, realize, oh, MUC1 is important, twist is important. We can just take those and just run flow cytometry then and do the same projections and it would still work. So those are, I know it's a little bit expanded answer, but yes, I'm hoping it will. But the only way to do that is to get on get a grant and get many patients enrolled, to be honest. So we'll see.
Thank you very much.
So, Loukia, I think your answer just shows your enthusiasm with the subject. So I had a question that's close to that. So how about, how is it, considering how expensive this method is, adding more markers more than immune? Because one of the issues is can you explain that heterogeneity that you have in your sample? So I guess other factors like epigenetic or metabolic will be playing a role. I don't know what's the chicken or the egg in those two, but
yes,
how would you address that?
Okay, so that's a very good point because when I talked a little bit about systems biology, I forgot to say something. It's using different approaches and studying the system, but it's also integrating data. So that's a big thing now. There's a big push to integrate different scale data. It takes a lot of computational analysis and platforms, but people are already trying to do it at least 2 or 3 at the same time. So for example, there is technologies that will give you at the same time all the single cell RNA, right? And then also some of the proteins. That's one way. So transcriptomic and proteomic. There's ways to integrate the imaging with the CyTOF. Since you have the same clusters, because you use the same panel, you can hypothetically layer on top and integrate that. Epigenomics, there are antibodies that probe or epigenetic markers. And actually, when I was at Stanford, there was one PhD student, that was her thing. She was building a panel of HDACs, and all that. Maybe the paper's not out yet. So like I said, there's capability to go up to 100 proteins, right? You can imagine once you do your own studies in your lab and you kind of know what's most important, you can build a panel and also have epigenetic markers and also have metabolic markers. Because at some point, yes, it's good to have everything, but you can't explain everything with everything. You have to nail it down to the most important. But there are ways to integrate and get to the bottom of it. Another way of you can think about, there's another imaging technology, where it's mass spec imaging. Now, in that case, you don't get the cells and their phenotypes, you get their metabolic products, right? So you can get their lipids and their metabolites, and you can layer that on top of the codex. So you see cell types, cell phenotypes, and cell metabolites. So again, it's a matter of, within your lab and your interests is you kind of know the biology, and then you build the most efficient way, of panels to interrogate these things at the same time. It's the curse of dimensionality. That's what they call it. We have the technologies now, right? There's a lot of technologies out there. The point is, how do you use them wisely, and how do you integrate it to get to the real biology? That's the problem now. And there are groups that are working on it, but we still have ways to go.
Okay, I don't know if there's any other question. I'll ask a few. So, another thing that came into my mind when you were explaining the lung cancer samples that you got, what did you use as controls? I mean, you have patients that are terminal. What would you contrast it to?
It's hard to, there's no control. So there's a technical control, right? So are you talking about the pleural effusions or the
the pleural effusion.
The only control, I could only run a technical control. So what I did is, while I was running the pleural effusions in the machine, I also ran my lung cancer cell lines to see if they were kind of similar and not like something completely different. And yeah, they had similar features with what I was seeing, but it's hard to say what is a control, because honestly, when I start doing the pleural effusions, there is no CyTOF studies on pleural effusion. So it's hard to compare. There are flow cytometry studies, but they were not focusing on everything. They were just looking at, for example, CD4 T cells, or maybe just the cytokeratin cells. So it's hard to say a control, and maybe that will be a feedback when we submit the paper. But the fact that it's clinical and you're going into that depth, I'm hoping that it will be, they would like it. But yeah, what is a control? I don't know what a control would be. So
definitely that would be.
I will say one drawback to the pleural effusions. I presented to everyone here today. It's just protein. So yes, there's a probability that some of those cells are not malignant, and we have to say that, because they're cytokeratin positive, but with the antibodies, I can't probe for mutations. So, the fact that I'm separating cytokeratin from CD45, it's good, but it's not perfect. Now, I did see studies that actually, in the field of circulating tumor cells, there's applications. That's what they do. They take out CD45 and they take EpCAM, and then they'll confirm if they are if they have mutations or not. But that's the first step. That's how you first separate a tumor cell from the circulation. And there are markers that would separate normal lung cells that might be floating in the pleural effusion. And that's something that we would have to work on. But yeah, other than that, not many controls.
But you could have like a normal lung cell that
yes,
transition of becoming bad,
you
right?
could. Yeah,
okay.
you could.
Okay.
True.
And along those lines, you mentioned something about solid tumors, and I guess the question is, can you get you have to have fresh samples to do your technique? I guess you cannot use fix.
For CyTOF, yes, they have to be. I would stay in the lab. Sometimes so the doctor would call us, the assistant. There's going to be surgery today, and because it's the clinic and things happen, I would have to stay until night until the tumor was out. Pick it up and go and immediately dissociate, count the cells, make sure they're viable, and then fix and freeze. This is where the imaging is a little bit better, because you can just do it on fixed samples. And you get the same answers plus their architecture, right? Okay. But it is more expensive. It's even more expensive than CyTOF. And don't forget that, when you get slides to stain, you only get one chunk of it, right?
Yeah.
Everybody knows that the tumor might be very different all the way around, but still, there's so much data that come even from that slide that it offers immense information in new biology.
Excellent. So we have a question from Dr. Andreas Hajikambis. Andreas, you're ready to go.
Hello, can you hear me?
Yep.
Yes.
So I would like to congratulate Loukia for this excellent, fantastic presentation. Thank you very much for this.
Thank you.
And I would also like to thank our coordinator of this series of lectures, Maria Charlie. Maria is doing a very good work in preparing all this. So thank you very much, both for the presentation, Loukia, and Maria for this organization. Thank you.
Thank you. And I apologize, Loukia. I didn't mention that Dr. Hajigamdis is the president of the Biological Society. Sorry, I'm very awkward yet as a host, but please forgive me.
And I just want to say again, thank you for having me. This is an honor to be here and to meet everyone. And I do thank Maria too. It's great to see her, even through Zoom.
It would be a great miss if I didn't have you. And as Dr. Delta has noted, we need more Cypriot scientists coming up and showing the rest of Cyprus what we do outside Cyprus, and be beacons for young scientists and people that want to.
True. Thank you.
Okay.
And
so,
if I can say one more word,
yeah,
I would like to invite also Loukia to become an associate member of our society.
Oh, thank
it
you.
will be a pleasure to have you abroad, to be one of our members.
I would love that. Thank you very much. I appreciate it.
Thank you.
Thank you, Adele. So let's see. Does anybody have another question? Loukia, I have to give you another question. So I think you mostly address the factors that limit the technique, or your CyTOF technique. I guess it's the status of the sample and the expensiveness. Are there any other limitations to this?
Well, I mean, obviously, when you're doing the CyTOF, like I said, you have to dissociate. So there's a lot of signaling that gets affected. And there's also some cell types that are more sensitive to dissociation than others. So you tend to lose a lot of the fibroblasts, you lose a lot of myeloid cells. So there's literature out there that kind of have studied. So yeah, you have to take that with a grain of salt, that something is not, it's not 100%. But again, there's so many cells you're analyzing, so many markers that, you're close to something important anyway. But yes, that's one of the limitations. But, other than, and then, again, it's only protein. Like I said, there's technologies out there that are trying to link transcriptomic with proteomic data. But it is a very cool technique, and it's changed single cell biology at the protein level. And but like I said, now there's more and more of a push now to the imaging aspect, because again, you get the same answers, but then you can do fancy neighborhood analysis. So if you can do that, why even break down the tumor? Now, in the case of pleural effusions,
you need that.
You just do the CyTOF. Yeah.
And do we know if there are any startups that are trying to bring it to a more close to the clinical side, like less expensive, so it's more applicable to outside of the lab, I guess? I know it's new.
I mean, I'm sure I'm not aware. What I think what's the case is that there are big grants that try to do that, but whether it's established or not, I can't tell. So for example, here at MD Anderson, there's a huge CyTOF imaging core, but I don't know if it's actually used. It's probably used under a grant to answer a question, but whether for diagnostics or not, I don't know. So I don't know the answer to that. But I would at some point it's going to get there. But like I said, if you can then, we scale up, but then if you find important proteins, then you go back down and you get the same answer. I don't know. In terms of biology though, and seeing things that you never realized, it's unparalleled, right?
Yeah, so, it's amazing. I mean, it's a technique that allows you to see beyond normal limits, like even just the number of even 30, having 30 proteins at the same time. That's crazy.
It's an immense amount of data, even 30, and people do it with more now. And because I'm a protein person, I was a protein person all my life. you know, everybody does single cell RNA-seq, but, you know, and many times it's not translated. It doesn't give you the function. It gives you a very nice whole characterization, but if you don't see it at the protein and it's not targeted.
Exactly. Yeah,
you know.
yeah. You need the systems approach.
Yeah, exactly.
Exactly.
Yeah.
So yeah, that's I'm sure we'll see you in the future of systems biology approach.
Hopefully.
CyTOF and codex and everything included in between.
Hopefully.
So, yeah, if we don't have any further questions, I think I gave most of my questions to Loukia.
That's
So
okay.
I would like to thank everybody for joining us today. Thanks to Loukia, who gave this wonderful talk and educated us on the subject. And yeah, we look forward to seeing you in the next talks. And thank you, Loukia, again.
Thank you very much for having me.
It was a great honor for us too. Thank you. Have a good afternoon, evening, whatever you
morning for us.
Good morning then. Bye-bye.